Good evening, everyone, and welcome to our webinar. So, today's webinar is hosted by Mr. Ansa CK, who is currently working in AIMS CAPFIMS Delhi, and he was former assistant professor in MES Medical College, that is in Malappuram district in Kerala. So, we are warmly inviting all of you to our webinar, and I hope everyone will enjoy the session. So, Answer, sir, you can start the session right now.
Hello, am I audible?
Yes, sir, you're audible.
Okay. Okay, so good evening, everyone. So thank you for introduction and also for the SimplyMED and Medline Julian Academy for inviting me for this session. Okay, so let us move on to our session. It's a great privilege to speak with you all today on a topic that has transformed a landscape of biomedical and analytical science. And this instrument, this advanced analytical technique that has a revolutionized and rapid identification in microbiology and the beyond what is in the message. Okay, fine. So it is an advanced analytical technique that we can revolutionize. We can use as a revolutionizer for the rapid identification in the field of microbiology and also for the another fields also. And with the increasing demands for the accuracy and the speed in the diagnostics, the MALDI-TOF has emerged as a cornerstone of modern laboratory practice. Okay, so that's why I am talking about the MALDI-TOF. And before starting this session, actually this MALDI-TOF technology is introduced in our labs or our hospitals. Most of the labs, they are introduced this technology within 4 to 5 years. Most of the labs, okay. Some other hospitals are already installed all these things and very routinely they are going on that instrument. But actually, you know that this instrument or this technology is developed in the 90s, early 90s itself. But we took some time to implement this is for our diagnostic purpose. So that's why maybe some people's not more aware about this instrument. Maybe some of the peoples are currently working on this instrument. I don't know. So in this session, I would like to deal a small little in-depth knowledge on these all kind of things from the basic principle to some kind of interpretation and as well as the benefit limitations and some other recent research studies undergoing on these things, all things I have discussing in this session. So we can move on to this session. Okay, so before starting, a small disclaimer is that this presentation is for only for educational purposes only and does not promote or endorse any specific brand or product. And here you can see some of the newspaper cuttings that is showing that some deaths occurred due to the some bacterial infections or other microbial infections. So all these deaths may be occurred due to the late diagnosis or the some delay in the reporting. So in some of the cases, we can save the some patient's life with the early diagnosis. Please mute the participants. Hello, please mute the participants. All of you, please mute. Okay, so thank you. Okay, so these all deaths we can reduce by implementing some kind of identification instruments with a very less turnaround time. So that's all of the basic importance of these kind of early identification systems. Nowadays, we are using some other rapid identification systems like Vitek, but Vitek has also some other limitations because that also we need some time around 15 hours after the isolated colony. Okay, so that also may lead to some other late diagnosis. So all these things denoting that we need a early diagnostic instruments for the better medical diagnostic field. And we are in an era where rapid, accurate, and high throughput technologies are not just desirable, but absolutely essential one. Whether we are trying to identify unknown bacterial pathogens or analyze some kind of proteins, proteomics, and also to detect some disease biomarkers, we need a tool that are both reliable and efficient. So that kind of instrument is our MALDI-TOF. Here it is some of the studies that are undergoing on the some researches on the MALDI instruments. So while implementing an instrument to a medical field, we definitely want to know what are the consequences or what are the results or what are the outcomes of that implementation. So here some of the studies, now also some of the studies are undergoing on for the evaluation of this MALDI-TOF or for the diagnostic purpose. Okay, so this is a already not an emerging technology, but it is already emerged in some countries. But in countries like India, that is installed in very few numbers of hospitals. Okay, so that's why it is an emerging technology. Okay, so that is MALDI-TOF MS. You can say that MALDI-TOF MS is the abbreviation form of Matrix-Assisted Laser Desorption Ionization Time-of-Flight Mass Spectrometry. Okay, so actually the principle and all much of things included in this name itself. Okay, so we can call it as MALDI. Okay, with a nickname as a MALDI. Okay, so basically it is matrix-assisted laser desorption ionization time-of-flight mass spectrometry. Okay, so here it is a picture that's showing during my lab life I have used to conventional methods for the identification of bacteria. So here I was used for so many kind of culture media, different types of biochemical media and different types of tubes, media and all things for a species-wise identification. All of you know that the difficulty of the inoculation and interpretation, making of media, even the washing of media, all kinds of workload you have now. But in this case, and also some more space also needed. Okay, more space also needed for
type
please mute. So this kind of analysis conventional methods needs a huge time and as well as huge spaces are also needed. So we can reduce please mute all. So this kind of convention, this kind of hectic process we can reduce by using MALDI-TOF. Okay. So in this session, I would like to discuss some little in-depth knowledge on the basis of beginning with its principle and history, small history, and workflow of MALDI-TOF, and also the components, the essential components, and also some other optional components are also needed, and some sample preparation process, some little you can consider it as a online workshop like that. Okay, so sample preparations and what are the benefits of MALDI and what are the limitations of MALDI, just like that. Okay, so first of all, you have to know the principle. If you are going to study any kind of instrument, if it is MALDI-TOF, Vitek, or any other instrument, you definitely know the principles. Okay, so what are the principle of this MALDI-TOF? That is basically mass spectrometry. Mass spectrometry is a wide branch of topic subject in the physics. It is basically mass spectrometry. Mass, you know mass. Okay, so spectrometry means, spectrum means a spectrum. Spectrum means a graph. Okay, graphical representation. We can call it as spectrum. And metry means measurement. Okay, metry means measurement. So mass spectrometry means it is something measuring with the help of spectrum. Okay, something measuring with the help of spectrum. Spectrum means graph. and it involves some kind of masses also. Okay, so mass also involved in this kind of measurement. So in briefly, we can say that it is a technique, it is an analytical technique in which chemical compounds are ionized into charged molecules and ratio of their masses to charge is measured. Simply, we can say that if a molecule is ionized into different ions, each ions have their own mass and charges. Okay, so these ions will move through a vacuum tube or any surface or any space according to their mass. A smaller mass ions will move faster and heavier mass ions will move slower. That is, the equation of velocity or the time is also related to that. So that is time of flight. Okay, so smaller mass ions will move faster. We know the kinetic energy half mv square. Kinetic energy is equal to half mv square. So if a small mass, it will move faster. And also, if a small mass, it will need very less time. Okay, so just like that, we can these applications these principles we can apply into the MALDI-TOF instruments. This is the basic principle of the MALDI-TOF. Okay, MALDI-TOF MS. Okay, here it is. I'm showing a video. Then you can easily get a picture on this principle. Okay. Okay, so that is the principle. I will explain once more. Before that, there is a small timeline. Nowadays, we are using MALDI-TOF in our labs, but it is actually developed on the early 90s itself. Mass spectrometry is the principle was discovered by Francis W. Aston. And after that, so many other scientists are developing different kinds of modification on these techniques. And in 1995, Harris and Hillenkamp, two scientists, they developed or they coined the name MALDI. And after the 1990s, the MALDI was used for the identification of bacteria. And recently, 2002, Yoichi Tanaka is a scientist who got the Nobel Prize for the developments in the MALDI. So many kind of things are undergoing on the MALDI now itself. And these are the two scientists, that is Hillenkamp and Karas. Please mute all. 1 second. And here, what's happening inside the MALDI? Okay, MALDI. Yes. Okay, so what happening inside a MALDI-TOF? Okay, so basically we need a target plate. Target plate is nothing but a solid surface. Okay, a solid surface, that is a plastic coated, metallic coated a plate, that is a solid surface. And we will apply our sample onto that target plate. Our samples, what is our sample here? We can apply sample as, if assume that you got a urine sample and you want to identify the organism in the species level from that urine, first of all, you have to inoculate that urine to as per the appropriate protocol on the definitive culture plate. Okay, so if you inoculated the urine sample today, then you will get a culture on tomorrow or overnight culture. And after the overnight cultivation, you will get a isolated colony. Then you should pick that isolated colony. That isolated colony is our sample here. Okay, you just note that, not our sample directly. Here it is, our sample is that the isolated colony is. If it is any contamination or polymicrobial colonies are there, then we will definitely not get the result. So we need a isolated colony. So that isolated colony is the sample for the MALDI-TOF. And we apply that sample to this target plate, our colony into the target plate. That sample is maybe changing according to our procedure. I am telling about the microbial identification. If it is some kind of proteomics, protein analysis, we have to use another samples. Okay, so you just apply that colony on this target plate and then add a matrix. Matrix is nothing but a chemical solution which is useful for the energy absorption. Okay, so energy absorption and as well as it will help him for the ionization also. Okay, so what is the need for energy absorption? It will act as a outer covering for our sample. Just like that, here you can see here a sample, and it's a green color, that is the matrix, and a laser beam is here. This laser beam will be pulses this sample, this molecule, this analyte into different ions. Okay, the laser will pulses this analyte to different ion particles. So this ionization will be takes place according to the laser beam. So we know that laser beam have some kind of energy. So that high energy laser beam will be pulses on this sample. It may lead to some damage to the sample. So to reduce that damage, to reduce that energy, we need a absorption material. That is basically matrix. Okay, so the matrix will be reduce the impact of laser beam on that analyte and also helpful for the ionization with the laser. Okay, so this analyte will be ionized into the different molecule, different ions. We know that different ions have their own masses. The small mass ions may be there, heavier mass ions may be there, like that. So these different mass ions will disperse from this analyte, and here it is some kind of electric magnetic fields are there. So according to that electric magnetic field, it will move through a vacuum tube. Okay, vacuum tube, that is known as flight tube. Okay, flight tube. So these all ions will move through this flight tube or our vacuum tube according to their mass, because it is basically working under the principle of mass spectrometry. Okay, so I'm already told that the ions will move according to their mass. The lighter mass molecules, lighter mass ions will move faster, and heavier mass ions will move very slower. So according to that, it will move through this flight tube. And we have a detector on the top of this flight tube. And this detector will detect all ions according to their mass, or according to their time. Okay, so from this we can say that the lighter molecules, the small mass ions will move faster, and it will needs only less time. So according to that, this detector will be detect or sense the detection, the beating of these ions, and it will lead to a spectrum formation. Okay, here we got a test spectrum. Okay, the spectrum of mass spectrometry of our analyte or our sample. Here we got the spectrum. This is actually MALDI. Okay, so some of you can have any other doubts, because from this spectrum we can say that it is a Staphylococcus aureus or it is a E. coli or anything like that. That is our software. we have already input some kind of databases in our software. That is, Staphylococcus aureus have this kind of spectrum. Escherichia coli has this kind of spectrum. Just like that, 2000, about 2000 spectrums are inside a database. So that database already installed in our software. So our spectrum, this test spectrum will compare with the database spectrum. Okay, I think you understand. Okay, so this test spectrum will compare with our database spectrum. So from that we can say that it is, if it is matches with any other database spectrum, we can say that organism. If this spectrum is matches with Staphylococcus aureus, the machine will say that it is Staphylococcus aureus. If it is matches with Salmonella typhi, the instrument will say that it is Salmonella typhi, like that. Okay, so this is the basic principle or the basic workflow of the MALDI-TOF instrument for the microbial identification. Okay, all these comparison, database comparisons, all are occur under that software without any manual interpretation. We will get. Okay, with the confidence value itself. Okay, approximately above 95% or 90% confidence value we are taking for the better result. Okay, so this is the basic principle of what's happening inside the MALDI-TOF is. Okay, and so many kinds of brands are providing MALDI instruments. Here it is, I am showing some kind of instruments that are basically using in the different countries. The Bruker biotype are mainly using in the US, America, and Biometrics White MS is mainly using the Indian states, and Render, M-Discover, Shimadzu, Axima, Hardy, Autobio, and so many kind of different kinds of MALDI-TOF instruments are using different countries. So hereby, I am mainly discussing on the Biometrics White MS instrument. Okay, all are working under the same principle, but the some kind of the material components and the consumables may be different. Okay, so here it is the essential components which needed for the MALDI-TOF instrument. Okay, now we have already discussed about the principle. Okay, I hope all of you understand the principle. And next, I am move on to the one by one step of MALDI-TOF. Okay, the small workflow. How will you do MALDI? Okay, if you get a chance to do a MALDI-TOF experiment, MALDI-TOF identification, then how will you do? Okay, so for a workup, for a protocol, we need essential components as these much things: metrics, target slide, disposable loop, calibrators. These all are the essential components for the MALDI instrument. Okay, so and all other some optional requirements are also. It is depends upon our what is our identification organism. It may be a bacteria, yeast, mold, MTB, like that. So depends upon that we need some other optional requirements also that are given here. And we are going to the essential components one by one. We can go. So first one is the target slide. What is target slide? Target slide is nothing but a solid surface where we have applied the sample. It is also the design of target slide is different, the varying according to the brand. The first picture that's showing that this is a target slide of Biometrics White MS. Hello. Yes. This is the first one is the target slide. This picture that's showing the first one is the target slide of Biometrics White MS MALDI-TOF instrument. And second one and this third one is all or other kind of instruments. Target slides we are using for the other kind of instruments. Okay. Some Bruker and applied biosystems like that. So here I am focusing on the first type. It have a 48 sample spot. Sample spot means the spot or the place where we have to apply the sample or where we have to apply the colony. That is the sample spot. Here in one slide, we have a 48 samples. This 48 sample spot, this total 48 sample spots are divided into 3 groups. Okay, they are divided into 3 groups as per this one. Okay, so we will get 16 spot on each group. Okay, 16 sample spot on each group. You can see from the slide. Okay, so this kind of 3 groups are present in the target slide of this kind of Biometrics Vitek MS multi-instrument. So each group consists of 16 sample spot. And also another major thing is that each group have a calibrator spot also. A small spot in between this sample spot. You can see that. Okay, here it is highlighted with a yellow color. This is the calibrator spot. So one slide consists of 48 sample spot with 3 calibrator spot. Okay, 48 sample spot with 3 calibrator spot. So every group it need a calibrator spot. That means if we are doing any experiment with this set, this group, if you have 1 sample to identify, or if you have 4 samples to identify, if you have 15 samples to identify, if you have 16 samples to identify, anyway, you have to use this calibrator spot. Okay, if you done the test with only 1 or 2 samples, then you can't use it anymore. Okay, this group you can't use anymore. That is the problem. Okay, so better to use with 16 samples. But you can use these groups otherwise. Okay, but this group, one group we can use at a time only. And next time this group we can use, and next time we can this group. So 3 times we can use one slide, but one group you can use only one time. Okay, that is the major problem of this instrument. Okay, because if you have only 1 sample or if you have only 2 samples you have to process, then you have to use only this spot. These all 14 spots are remaining, but we can't use it anymore. Okay, that is the problem. Okay, some other reusable type target slides are also available, but it is a little bit costly, and the result validation is also something difficult. So that's why this disposable target slides are mainly using. Okay, so that is the thing about the target slide. And next essential consumable, that is matrix. Okay, matrix. Matrix is nothing but a chemical compound which is mainly, I have already told that it is mainly using for the energy absorbent. Okay, energy absorbent. Okay, it is a crystallized molecule. Okay, so it is basically different types of matrices are available. Commonly, we are using CHCA, that is alpha cyano 4-hydroxycinnamic acid, cinnamonic acid, 2,5-dihydroxybenzoic acid, like that. Okay, and next essential consumable, that is calibrator. It is very important. It is very mandatory for each running. Okay, so in previous slide, I have already told that there is a calibrator spot for each group. So that calibrator spot, we have to apply this calibrator. Calibrator is nothing but E. coli ATCC 8739. Okay, we know that some other E. coli 257 and 224 ABST, like that. Here we are using E. coli ATCC 8739 for the as the calibrator for the multi. Okay, and another major thing is that if this this strain is contaminated or it contain other polymicrobial organisms also, so then you will definitely not get the result. That is the basic problem. And another thing is that first, our instrument, we are loading with the sample spot with the sample and calibrator, and the first machine will be first looking to the ATC system or the calibrator. If there is any contamination or any other problem for your calibrator, then the machine will not read your samples. So that's why the calibrator maintenance is very important. Okay, calibrator strain maintenance for MALDI-TOF is a very important thing. Okay, if there is any contamination or if there is any other polymicrobial organisms contained this culture plate, they'll also affect our the result. Okay, so that is the very important thing. And we can use other calibrators also, Enterobacter aerogenes ATCC 13048, Candida glabrata ATCC MYA 2950, like that. Okay, depends upon the protocol. And another thing is that is disposable plastic loop. It is the very thing. Okay, because if we are using metal wire loop, it will get scratches on the slide. That is something metal coated like that is disposable, sorry, stainless steel like that coated type of slide is that. So that's why we are using a metal wire loop. It will scratches that sample spot. So it will be affect our result also. So that's why we definitely go for any disposable wire loop. And we can avoid the contaminations also. Okay, it will improve the accuracy. But nowadays, in most of the labs, we are not using this kind of disposable plastic loop. We are using like toothpick or like that, any other sticks also. That's enough. Okay, that's all are good. And another thing is that we have already told that 48 samples we can apply on a single slide. So at a time, we can load 4 slides. Okay, 4 slides we can load at a time with a slide adapter. So total 192 samples we can load into a instrument, okay, at a time. And also this is a workflow of a multi, that is we have to first sample preparation needed and make a spot on the slide and add metrics on the spot and then place on slide adapter, load into instrument and spectrum analysis and reporting. Very less manpower needed on this technique. That is the very important thing. If it is we are using Whitec, we have to use some other manual procedures also there. We have to suspension preparation and optical density measurement like that. But here it is very less manual interpretations, manual procedures are needed. Okay. And also this workflow is also something different according to the identification. We can use the MALDI-TOF for the bacteria identification, yeast identification, mold identification, mycobacterium, nocardia, blood culture, etc., etc. Okay, different kind of technologies are developing day by day. Okay, so nowadays we are mainly using for this kind of identification. So first, I would like to tell about the bacterial identification. It is very, very important. So these all are the major consumables needed for the bacterial identification by the MALDI instrument. So what is the sample here? If you have to identify a sample, if you have to identify a microbial or bacteria from a blood or any other urea or any other sample, you have to culture in a specific culture media. Okay, so these culture media we can use. These kind of culture media we can use. This is all are mainly routinely we are using
Okay.
type of
Bones
culture.
and muscles of our body work together to help.
Please mute all. Okay, so these kind of cultural media we can be used. Okay, so these all are the recommended media by the instrument. So after that, the cultivation, the isolated colony will pick up with the disposable wire loop or any other plastic wire loop or plastic stick or matchstick, that wooden part or any other toothpick like that. We can just pick the isolated colony and smear on the target slide. And after the smearing, add matrix and keep it for dry and just load into the Vitek MS instrument. It is the very, very small process. Okay, when you just see the image, we have to work only these kind of things only for the bacterial identification. If you just look into the protocol for the conventional identification, for a species-wise identification, you have to do what kind of jobs? You have to inoculate into the different kinds of biochemical media. You have to microscopic the grams, different kinds of microscopic examinations needed. And then that kind of different procedures are you had to do, and so many time is needed. But here, very less time needed and very less labor-intensive procedure. Okay, this is the very important thing of the MALDI-TOF. And this is a video that's showing the bacterial identification. Okay, just go through this video. Okay, so this kind of very little procedure, we will get the result. Okay, just like that, Staphylococcus aureus with a confidence value of 99 level. Okay, Staphylococcus aureus and Staphylococcus maltophilia, like that, Stenotrophomonas maltophilia, Pseudomonas aerogenes, like that. This is all are given some examples here. So that's like that. We will get easily, we will get the result very easily with very less labor-intensive procedure. These all are some instruments, and this is a target slide which we are applying with a small plastic applicator stick like that. And here it is the basic software interface of this instrument. And next thing is that is yeast identification. For the bacterial identification, we already discussed some few procedures. And for the yeast identification, we know that yeast cell is more more complex than this bacterial cell. So that's why we need some more step. Some more step is not only one step is additional. That is formic acid addition. Okay, so for the yeast identification, we have to usually use in this kind of culture media. There is chrome agar or SDA, sabrodextrose agar or potash dextrose agar, like that. And for the protocol, we have to just one additional step is there. By picking the colony, smear on the target state, just you add 1 microliter or 0.5 microliter of formic acid and dry it. Okay, and after that, add matrix. Okay, only one additional step is there for the yeast identification. If you are suspecting any, if you have got any creamy colonies on SDA, you have to go through this protocol. Okay, just like that. So for yeast identification, only one additional step is needed, formic acid addition step. Okay, and all other procedures are as similar to bacterial identification. But for the mold identification, we need some other extra procedures, some other extraction-like procedures are needed. It is more, much complex process. Okay, mold identification and Mycobacterium tuberculosis and Nocardia identification need some more complex procedures as compared to bacteria and fungus. Not much complex process, but also some processes are needed. So that's why we need another kind of optional, the materials also needed, the reagents like ethanol, acetonitrile, formic acid, like that also. Okay, so here we need some other protocol like a extraction like procedure. We have to wet a sterile cotton swab using a APA suspension media or sterile deionized water, and then collect the mold from the agar plate using a moistened swab. And we will suspend the collected material in a tube containing 900 microliter of 70% ethanol and mix that tube using a vortex. And after that, centrifuge the tube to create a pellet. And after that, remove and discard all the supernatant, and to that pellet we have to add 40 microliter 70% formic acid, and then add 100% acetonitrile, and then again centrifuge and create a pellet. And that pellet, this finally we have prepared a pellet. That pellet is we are using as a sample here. In the case of bacteria and fungus, we have directly picked the colony and applied into the target slide. Here we have applied this pellet into the sample slide. Target slide. Okay, this pellet is here our sample. Okay, so this is the basic importance of the mold identification. Okay, we need some other complex procedures, complex steps are also needed. So that's why in the most of the cases, mold identification and MTB identifications are not identified by the MALDI-TOF. And for the mycobacteria and nocardia also, we need some kind of other complex processes like this one. Complex processes like this nocardia identification. Okay. And here, when for the mycobacteria identification, we need some kind of media like LJ medium, middle group agar, like that. And for the nocardia also, we need some kind of BCYA agar, Columbia agar, like that. Okay, we know that nocardia is also give growth in the SDAs also. And for the next thing, Mycobacterium nocardia, this kind of some extraction procedure needed, like 500 microliter of 70% ethanol added to a glass bead containing tube. That means it needs some mixing, homogenization. So that's why we need some glass bead containing tubes for the better mixing, because it is more harder than the other bacteria and all other things. So that's why we need this kind of extraction procedure before applying to the target slide. And we have to centrifuge to create pellet and remove and discard all the supernatant, add 10 microliter of 70% formic acid, then acetonitrile, we again centrifuge and then create the pellet. And this pellet is using for the sample. Okay, just like that, a mold identification. Okay, so there is also some differences in the procedure. Okay, that is I just given an outline. Okay. So mainly we are focusing on the bacterial identification and the yeast identification, because mold identification and Mycobacterium tuberculosis or nocardia identification need some other pre-sample treatment processing, like this kind of extraction procedure we have to work. Okay, so that is the basic important thing. And here it is some case scenario. I am just showing that for some examples. A 54-year-old ICU patient with uncontrolled diabetes and CKD was admitted with a septic shock. On day 2, he developed a persistent high-grade fever, unresponsive to broad-spectrum antibiotics. Blood culture showed yeast-like growth, but species identification was pending. Despite empiric fluconazole therapy and catheter removal, his condition worsened and he died on day 4 of admission due to suspected invasive fungal infections. In this case, within 4 days, a patient got expired due to these invasive fungal infections. So usually candemia or the candida infections have very rapid death will be not occurring commonly, not occur. But most of the cases, we all are neglecting the identification of candida. In most of the labs, they are using candida albicans or non-albicans candidas. That two categories are using. But actually, we also give some importance to candida species also. In this case, we given the patient antifungal as fluconazole, but the patient is died on the after day 4. So by the conventional method, we got that on the candida differential agar, a creamy pink colony. This is look like a parapsilosis glabrata like that. We cannot confirm with a candida differential agar if it is a glabrata or parapsilosis like that. But we can say that it is a non-albican candida. So that's why we commonly given fluconazole as the antifungal. But after the death of patient, just investigated the things, and again that isolated colony given for the MALDI identification, we got it is Candida auris. Candida auris is a critical pathogen on that time. So if we got this result within that time, within the admission time or within the hospitalization time, we can change the antifungals to caspofungin, echinocandins like caspofungin or bicafungin like that. We can maybe we can we can give a good treatment to that patient. Maybe. Okay, so this is also a example for a late diagnosis. Okay, and here it is a picture that's showing a candida differential media, a green color colony on a candida differential media. Okay, if you have any suggestions by looking to this colony, anyone can say what your assumption by seeing this colony, which kind of candida? Stropicalis, Cosseri, any other? Usually candida differential media containing green color colonies, what kind of cruci? Usually on most of the cases is candida albicans. Most of it is also varying up according to the culture media, okay, brand of the culture media. Usually we are using the high media culture media, so that's why the candida differential media that will give candida albicans has the green color colony. Okay, so we can give it as a candida albicans with this candida differential media itself. But there is also some differences are there. It is something more hard to pick. So that's why we given it for the MALDI-TOF identification. So that time we got it is Candida ciferri. Candida ciferri is a rare species of Candida species. So these all species wise identification we have got by only by the MALDI-TOF. So that is the importance of the MALDI in our clinical impact. Okay. So here it is some other applications of MALDI-TOF are given: pathogen identification, antimicrobial resistance studies, mutation studies, epidemiology of infections, and some other protein peptide analysis, synthetic polymer industry, cancer diagnosis, and very wide range of applications for MALDI-TOF. And now I have already discussed some kind of technical aspect. Now I would like to discuss some other benefits of MALDI-TOF based upon the evidence, okay, based upon the several research studies. Okay, so what are the benefits of MALDI-TOF? We can just look. Here it is a study that showing that a comparative study between biochemical identification, that is our conventional identification, with MALDI-TOF identification. Here you can see that the mortality rate is only 6.1% by the MALDI-TOF identification. Okay, by the conventional identification, it is 9.6. So we can reduce 3% of mortality rate by the implementation of MALDI-TOF identification. Okay, so mortality rate we can reduce by the implementation of MALDI-TOF. This study telling that. Okay, MALDI can save many lives, but by early diagnosis and get start treatment immediately. And here it is a picture that's showing in my lab that is placed for the washing of these tubes. So this kind of washing, the tube washing is, you know that the washing of these tubes are very hectic procedure. And we can reduce this kind of laboratory waste also. If maybe it is disposable tubes or disposable culture plates, we have to dispose into the appropriate biomedical hazard bags. So that's also need a large amount of laboratory waste. And the active preparations of media and all things are also very difficult for the conventional identification. So we can reduce this kind of biomedical waste. Here it is another study that's showing that we can reduce the biomedical waste per month 6-fold. Okay, we can reduce the 6-fold of biomedical waste. Okay, so we can, by using the MALDI-TOF, we can reduce the biomedical waste 6-fold. Okay, according to this study. Okay, so this is also very important benefit of this MALDI-TOF. And also another thing is that is cost. Actually, the instrumentation cost, the capital cost is very high. You everyone know that. Okay, about crores and crores needed for the installation of MALDI-TOF. But the patient cost or the test cost is very less as compared to conventional method. It is a example given. If we identify a yeast with a conventional method, we need approximately 600 rupees as a test cost, not patient cost, test cost. Test cost is 600 rupees. But that same yeast we can identify with the species level by MALDI-TOF with only approximately 50 rupees. Okay, only 50 rupees we can identify that yeast very rapidly also. That is the cost efficient. Actually, MALDI-TOF is very cost efficient without considering any the capital cost. Okay, the lab test cost is very less. And here it is another study that's showing the cost reduction. So here we can reduce the cost approximately half of the conventional method. Okay, this is also very important for the use of MALDI-TOF. And we can lead to the improved clinical care and the reduction in the length of hospitalization. We everyone know that if you the increase in the hospitalization time, which leads to the so many other kind of problems. First one is patient cost. And also another thing is that emerging of the antimicrobial resistance. Is it? Okay, so if a patient is staying in the hospital more time, it will lead to the antimicrobial resistance. So by the reduction of the length of this hospitalization will definitely it will reduce the antimicrobial resistance also. In future days, antimicrobial resistance may become the critical infection of the critical type of disease among the patients. So we can reduce that antimicrobial resistance also. Here is another study that's showing that Usually the time of resistance, the time of antimicrobial resistance are starting from approximately 40 to 50 hours. So we can identify an organism within that time. Here it is showing that the MALDI-TOF identifications within that 40 to 50 hours. So that's why we can reduce the hospitalization time. It will lead to the reduction of the resistance also. This is also a study that is shown. And here it is another study that's showing the turnaround time. Okay, comparative study of conventional identification and as well as the MALDI-TOF identification. Here it is the comparison. Comparison means there is the test, the turnaround time comparison. Here it is in the conventional method, the Acinetobacter baumannii needs around 56.9 hours. Okay, approximately 56.9 hours needed. And by using MALDI-TOF, it is reduced to around half of the time, to only 28.8 hours. So we can reduce the turnaround time very much. Okay, here it is this table showing that this all bacteria have very less number of less time of turnaround time. Okay, all are significantly less turnaround time got by the MALDI-TOF. So these all studies are denoting that we can reduce the turnaround time, we can reduce the mortality rate, we can reduce the biomedical waste, and we can reduce the hospitalization time and all much things. Okay, so these all are the major benefits of the MALDI-TOF. It is a more reliable and small amount of biomass is required. Sample preparation is relatively simple. Reduction in the time to identification, more accurate, helps to quickly guide treatment decision, reduction in the length of hospitalization, a significant cost saving, reduction in the biomedical waste, etc. Okay. So many benefits are there, but also some minor limitations are also there. What are the minor limitations? First one is the very high capital cost. Very high capital cost. Please mute all. Very high capital cost. And also another major limitation is that the inherent similarities between some organisms. It will may lead to the misidentification. The important example for that is E. coli and Shigella. Because I'm already told that each bacteria have their own fingerprints, their own protein fingerprints. That is our that is their ribosomal proteins. So according to their protein fingerprints, we have analyzing the MALDI-TOF. So, but for the E. coli and Shigella, the both these bacteria have similar protein spectrum. Okay, so that's why most of the cases E. coli and Shigella will get as a misidentification. But also the newer updates, newer techniques, they are reducing this kind of misidentification, and new updates, it will very huge reduction in this misidentifications are seen. And also some for the Vitek, we can give the better result on the antibiotic sensitivity testing, but here we will not get any antibiotic sensitivity testing, but also we can detect some kind of antimicrobial resistance proteins, but that also under some kind of researches are ongoing. So this is also a limitation. Okay, but in future it may lead some other more updates on this field. On this MALDI, we can use for any samples, we can use for direct sample also in future maybe. And also we can use for the antimicrobial resistance detections, and also maybe some kind of artificial intelligence, machine learning may be combined with this MALDI-TOF. We can improve the interpretation and all things. Okay, that is maybe happening in the future. Okay. So these all are my references. So finally, in conclusion, the MALDI-TOF is more than a laboratory tool. It represents the power of interdisciplinary innovation, where chemistry, biology, physics, and all computations come together to solve real-world problems. Okay, whether it is enabling faster diagnosis in hospitals, unraveling the complexity of the human proteome, or pushing the boundaries of biomedical research, MALDI-TOF is a testament to how far science has come and how much further we can go. Okay, so thank you once again for your time, for attention. And now the time for you can you have any doubts, you can ask now. Okay. Okay, otherwise you can ask through my Gmail or WhatsApp, or scan by this QR code. Okay, anyway, you can ask. Okay, so I hope all of you clear. Okay, if there is any doubt, you can ask now. You can ask the questions by chat box or unmuting your mic like that. If there is any non-Malayalis or Malayalis, you can ask with your language also. Okay. If there is any Malayalis, you can ask in the Malayalam also.
Sir, can we do Y-Tech also like this when you're free, sir?
Okay, let us plan. Sir, can you arrange a session on Y-Tech to combat? Okay, let me try. Sir, can you share PPT? Yes, okay. Should we cross confirm with the colony morphology with each MALDI ID? Should we cross confirm with the colony morphology? Actually, MALDI is not much related to colony morphology. Okay. So if it is any colony morphology, we can get it to MALDI. Okay, but we cannot correlate with the MALDI identification. We can correlate with some other conventional methods, but also the sensitivity is very high. So that's why we can confirm the result according to the confidence value. Okay, if we go to a confidence value more than 90 or 94, we can definitely report the sample as per that. What are the technical errors with MALDI-TOF? Technical errors mainly one is that the polymicrobial growth. Okay, that is the major important technical error. Because if you have no isolated colony, you definitely not go for the MALDI. Okay, definitely you will get a most probably the result will be not come, and if there is anything that it will be misidentified. So the major thing is that you firstly observe the culture colony. That is only if it is, there is a isolated pure colony is there, then only you should do the MALDI. Okay, that is the major technical error. And other thing is there mainly the identification of this one, E. coli and Shigella. If you get any bacteria as E. coli, in the instrument itself, they are showing that there is a chances for the misidentification with the Shigella. Okay, but while confirming with the conventional, you can go for the E. coli or Shigella that. Okay, because E. coli and Shigella, we can easily identify by the conventional itself. We can easily differentiate with the conventional itself. And also other technical errors, I think this one, the software errors. Software errors may be a technical errors, but usually not much technical errors are usually not seen in the MALDI instrumentations. Mainly this kind of things. Cost of MALDI-TOF machines. Cost of MALDI-TOF machine is depends upon the brands. Okay, I think it is around 2 crores like that. Okay, that's also depend upon the brands. Okay. Sir, is Candida auris identification reliable by MALDI? Yes, Candida auris is the database is available in the MALDI-TOF, so that's why we can get the Candida auris. And also another thing is that if there is any critical pathogens are denoted by WHO, that will, according to the software, it will show this organism is critical pathogen. So in previous update of previous database of biomerics, they will show if we get any Candida auris, they will definitely the machine will be say that it is a critical pathogen. Okay, if we'll get a Cryptococcus Cryptococcus neoformans, they will show that it is a critical pathogen like that. Okay, so Candida auris is very easily identifiable by the MALDI-TOF. Can resistance pattern can be no a resistance pattern, no, we cannot measure the MAC or antibiotic sensitivity pattern, but we can identify the proteins, some kind of proteins responsible for the antimicrobial resistance. But also nowadays no much standard kits or standard protocols available for the resistance identification. In future definitely some kind of protocol will be come. What is the turnaround time? The approximately turnaround time is depends upon the sample load. Okay, so if we are using only one sample with one calibrator, we will get the result within 10 minutes or 20 minutes. Okay, so that 10 or 20 minutes needed only for the vacuum preparation. Once the vacuum preparation is completed, then it will give the result very rapidly. Okay, so another major important thing is that if we are loading around 20 or 30 samples, we will get the all result only after the complete this analysis. Okay, that is the major problem. We cannot get one by one result. Okay, we will get the result of complete reportings. That is the another problem. normal turnaround time for one sample is around 10 to 20 minutes. Okay. So you just see that we are identifying a colony, we are identifying a bacteria by species-wise identification by conventional method by taking 3 or 4 days. Okay, so here we will get within 10 or 20 minutes or within 24 hours. Okay, within 2 hours we will get the result. That is very important thing for the diagnostic field. Thank you so much. Okay, thank you. Will MALDI be the treat to future microbiologists in terms of job prospects? Threat to future microbiologists. Actually, the whole in all diagnostic field is another kind of talk. Okay, so it is in every kind of diagnostic field we have already implemented so many kind of automations. But why? But why many automations are not implemented in the microbiology field now itself? Because a manual interpretation is much more needed in the microbiology field. Okay, nowadays so many kind of total laboratory automations, total digital microbiology systems, total digital cameras are available for the colony interpretations. But also in that labs also we have using some kind of manual interpretations. So if we are implementing or if we are introducing new techniques, then also there is some kind of manual interpretations are needed. Okay, because all these kind of instruments not have any kind of knowledge as per a human being. So that's why it may not be a threat. Okay. Will the video will be available in YouTube? That you please contact the organizing team. Okay. And we will provide the cloud recording. Okay, okay. Okay, the organizing team will be provide the links for you. Thank you. If fresh isolates are required, it will still take time, around 2 times. That means if you have a sample in your hand, you need to cultivate it in the culture plate. If you like an example, if you have any urine sample to culture, so you just culture the urine in a blood agar and McConkie agar like that. So and after the overnight incubation, you will get a isolated colony. From that isolated colony, that fresh isolated colony, you take a isolated colony, pure isolated colony, and then process the thing. So after the 24-hour and plus 30 to 10 to 20 minutes, you will get the result. Okay, that means around 24-hour is needed for the cultivation. Okay, for the overnight cultivation, and after that 10 to 20 minutes. Okay, so that is around 1.5 days totally, we can say that. But actually turnaround times we are calculating from the isolated colony. So from the isolated colony, we need only 10 to 20 minutes. From the sample itself, we are calculating the turnaround time. It may be around 24 to around, if we are calculating around 18 hours incubation, then 18 plus 1 hour. That means within 19 hours or 20 hours, you will get the result. Okay, clear? All of you clear? There is any
What
other
is the purpose of calibrator?
Calibrator is the purpose means as like a quality control. We are using the if we are machine is working properly or other ways. Okay, so first we have to properly give the calibrator strain on that calibrator spot. Then the machine will be analyze that calibrator E. coli ATCC strain will giving same result. Then only it will be given our result. So that means it's like a quality control, like positive control, negative control we are giving in the all tests. So that's like a quality control checking was also given there. Okay. For that's why we are using the calibrator. Okay, clear? Is there any other doubts you can ask now? Otherwise, send your feedbacks, and if there is any doubts, you can contact me on this QR code or this mail or anything. Okay, so if there is any no doubt, you can conclude now.
Okay, sir. And before we conclude the session, I would like to thank the faculty, that is Ansar CK, for giving us your valuable time and sharing the most important or like the emerging technique that is like very useful for every microbiologist in every aspect. And I would also take some time to introduce this like our organization to you. And actually our organization is Medline. Medline is a professional hub that helps healthcare aspirants and professionals through every step of their GCC licensing procedures and ASAP certification process. Like we are providing these like documentation, data flow verification, and also exam booking and support throughout your licensing journey. Okay, and we have also coaching department, that is a coaching and learning division of Medline is SimplyMED Learning. And at SimplyMED Learning, we provide structured online coaching for a wide range of healthcare disciplines, including medical laboratory technology, radiography, physiotherapy, and anesthesia technology, etc. And also we have an online application which is called as SimplyMED Learning. Through that application, we are taking the classes. Okay, so if you want to know more about our application and our organization, please do give us a message. Okay, so I would once again thank all the participants who joined in our session, and have a great day. Thank you.